In a lot of the cases, the microneedles didn't form, or we had patches that were just too it was like Swiss cheese, there's a lot of holes in the product. Yeah. So that was the problem that we ran into. In fact, I mean, I might put it like that. Our formulation broke the method.
I reached out to you initially because I had seen you work in microneedles, and I've seen that you had done a lot of other research around microneedles, and there's been a lot of sort of side projects, if you can say. So thank you for joining me, and thank you for your time.
Thank you for inviting me.
The first thing I think we should start off with is really, if you can just tell me a bit about yourself. So what is your background of research? What is it that you focus on mostly?
I have a first degree in pharmacy. So I actually did an M.Pharm degree, and then I went on to do a PhD in drug delivery. And actually, my PhD project was on microneedle delivery of vaccines. That was a long time ago when that was, it still is quite a hot topic. After my PhD, I went to do a postdoc at Imperial College London, working on a project to do with critical care immunology. And that actually, a brief stint outside of pharmaceutical science actually opened my eyes up to, other possibilities. And then I kind of went back to pharmaceutical science and microneedles. And actually from that, I developed a research strand on diagnostic microneedles. So bringing in my kind of experience in critical care immunology, and also my, microneedle experience and expertise from my PhD study. And that's where it all began.
So let's talk a bit about microneedles then, because I've seen that, you've got a lot of work in that space. What are some of the major challenges that you're seeing in that space of microneedles?
Mm-hmm. I think with microneedles are a great technology for drug delivery. It solves a lot of issues that patients have. I mean, one problem one challenge that we often hear from patients is that a lot of people are afraid of needles, and we have spoken to patients, members of the public, and they like the idea of these needles being so small that they don't cause pain. So it can actually have a lot of applications in, drug delivery to treat a lot of diseases, especially in patients where a needle injection is problematic. So either the patients don't like injections, or you need injections so frequently that they become very, very invasive and intrusive. So there are many reasons. But with microneedles, one of the issues that we were trying to overcome was to reduce the dosing frequency. So the problem we're trying to solve was specifically how do we make a microneedle formulation that is basically like a platform technology that can release a drug for a month. So that was where we're kind of, what we were focusing on, and the problem that we're trying to solve.
Did you achieve 1 month, or what did you achieve with your sort of research?
Yeah, I mean, we did achieve 1 month, and actually we achieved 2 months.
Okay.
So the approach that we took was we had to take a different approach compared to what other people have done. And the approach that we took was we developed a composite material that actually combines both a water-soluble polymer and a non-water-soluble polymer. And the idea was that, this combination of polymers as a composite would partially dissolve, but part of it will remain for a long time, and remaining for a long time in the skin, which sustains the pathway for the molecule to move from the patch into the skin. So we did some work on that. The way that we did it was actually, in our polymer we have, as I said, 2 different types of polymer, and actually one of them is a cross-linked polymer, which forms a gel when it's hydrated. When water infiltrates, it forms a gel. So that kind of fits the prolonged release model. Within the gel is embedded the other polymer, which is water soluble. So actually, as the microneedle hydrates, the soluble polymer becomes wet and it dissolves, but it does so very slowly because it's kind of impeded by the other polymer which surrounds it. It's basically embedded in that network that's not water soluble. And after the soluble polymer dissolves, it then has to kind of diffuse out of the network that it's entrapped in. And again, it can do that, but it can do it only very slowly. And with the release of the dissolving or the soluble polymer, the drug gets released. And because the dissolving polymer is released very slowly, it's only dissolving very slowly and getting out of the microneedles very slowly, so is the drug. And that's how we kind of manage to sustain drug release for over 2 months.
What method of manufacture was you using to create these sort of multi-layered composite systems to produce the microneedles?
Mm-hmm. Yeah, so we actually started with the common solvent-casting method, which a lot of people use to make their either soluble microneedles or the hydrogel forming microneedles for prolonged release. So we started with that method. So that method is actually pretty straightforward in terms of actually doing the fabrication in the lab. Basically, you start with a mold, and you prepare your polymer in solution form, and then you pour the polymer solution into the mold and you dry it. And once it's dried, you eject the formed microneedle patch from the mold, and that is your product. So that has worked with a lot of formulations, and there's a lot of publications on this topic. So it was intuitive for us to try that method. So obviously with ours, we put our composite polymer solution into the mold, but we immediately ran into problems. Okay. The problem that we had was because of is brought on by the fact that our formulation is very viscous, and because of the cross-linking process as well for the insoluble part of the matrix. So that starts to kind of cross-link, in that manufacturing process. So our microneedle patches didn't turn out very well because in that process, actually, you put into the mold, and actually I missed one step, which is, you put it under usually either in a centrifuge to kind of force the liquid formulation into the very tiny cavities at the bottom of the mold, which eventually form the microneedles, or you put it in the vacuum to kind of force the air out from the mold. Now that step was the step that got messed up with our formulation because of how viscous our formulation was. We put the formulation under vacuum, but the air was not able to escape because it had to move through a very viscous polymer solution, which was not allowing it to pass through easily. So we ended up with a lot of failed kind of products with a lot of air entrapment in the polymer. And in a lot of the cases, the microneedles didn't form, or we had patches that were just too it was like Swiss cheese. There's a lot of holes in the product. Yeah. So that was the problem that we ran into.
What did you do after that? What did you stick to solvent casting? Did you adjust the method, or what was the next step after that?
Yeah, so we found out actually, the reason that the solvent casting, the traditional solvent casting method worked, for other people, was because they were using relatively low polymer concentrations. But in our formula, we had quite high, much higher polymer concentration, which made the formulation much more viscous. So we ran into this problem. So in fact, I might put it like that. Our formulation broke the method that worked for other people. Yeah. So we needed to adjust it, and we knew that the problem was with the we're removing the air after putting the liquid into the mold. So having put the liquid into the mold and trap the air in the mold and then trying to remove the air was the problematic step. So we needed to get rid of it. So then our approach was to actually, why don't we remove the air first and then put the polymer solution in? And that was the approach that we took. The issue with that was logistically it was difficult to do because in order to remove the air, to have removed the air first, and then put the polymer solution in while the air is not present, we need to do this in a vacuum chamber.
Yeah.
And there was no way for us to kind of manually handle this, equipment and the sample, the solution. There was no way for us to handle that manually in a vacuum chamber. So we developed a method which we called Vac and Fill. So the Vac and Fill basically means that you apply vacuum first and then you fill the mold with the polymer. So this is basically the other way around compared to the traditional method where you fill the mold first and you try to remove the air by applying vacuum. In terms of how we overcome manipulating these equipment and samples in a vacuum chamber, we'd actually leverage the fact that we're applying vacuum in order to automate that process, to some degree, so that we don't have to manually intervene when the equipment and the sample is under vacuum. And we made use of a very kind of basic principle that, if you have a container that contains air and you put that container in a vacuum chamber, the container will expand because the air in the container will expand. And in order to allow us to pour the polymer solution into the mold under vacuum without actually manually handling the equipment, we would make use of this principle. So we modified a traditional syringe. Yep. We put some air in the syringe. We put the liquid formulation in the syringe, and we drill a hole in the syringe as well. So, but we made it such that, initially we would assemble the syringe. We make sure that the air in the syringe is there and is the right amount. And then we will put the mold, position it under the hole that we have kind of drilled in the syringe. And we will apply vacuum, and the air that we have left in the syringe will expand, and it would push the plunger of the syringe. And by doing so, it will open up the hole, which would have been originally concealed. But when the air expands, it will open up the hole, and the liquid formulation will flow out of the hole and into the mold and fill the mold, all without kind of manual intervention from us. Now, we did have to calibrate this very carefully. I have to say, it was originally, as a concept, we were quite confident that it would work. But getting it to work reliably was a lot of hard work because it was a lot of calibration. We had to basically calibrate, the amount of air that we put into the syringe. We had to also calibrate it, so that the hole is open at the correct pressure as well. So we don't want to open up the hole too early or too late. And also found out there are some things that you just never thought about. Things like if you use a different syringe, you will have a different mechanics, which will cause your mold filling process to be different again. So all of these parameters needed to be calibrated. So we did that, and we basically made sure that it worked for the range of polymer concentrations. So we kind of did a study to look at different viscosities of polymers and to kind of study how they failed using this method under vacuum. So yeah, we were pretty happy that we could apply this actually to a range of different polymer solutions, different viscosities, different formulations. So with this, we solved actually the problem with the air entrapment in our kind of solvent-casting method. We still use solvent-casting, but it is a kind of slightly modified kind of sequence of events.
Yeah, no, I think it was really interesting to see that you did the opposite of what was conventionally done. So instead of fill and vac, you went in the opposite direction, because with fill and vac, as you say, once the air is trapped in there, you're kind of having to then work backwards to get it to the standard that you need. Whereas with vac and fill, you're kind of building in the quality in the way you're doing it, because you're just removing the air and operating under a vacuum essentially. So then you're preventing that air from sticking into your needles. What was the general quality like of the needles in terms of, were they mechanically good? Did you test the drug release? How did they turn out?
Yeah, they turn out great. So we, in terms of mechanically, they're strong. I mean, we started, when we first deployed this Vac and Fill method, we started with quite a low bar actually. So we were aiming to just form a microneedle patch with all the needles being present. Because previously with the traditional solvent-casting method, we were getting products that had no microneedles at all, or there was like Swiss cheese, so we had a lot of bubbles but no needles. So you have to kind of understand that we was starting from that position. So our first aim was to actually just form microneedle patches with all the microneedles that should be there, making sure they are all there, and that was it was achieved pretty quickly. And then once we got that to that point, then we started looking at how strong the needles were they able to penetrate skin as intended. So we did all this kind of like standard basically quality control tests for our needles, and they perform very well. So if we have our benchmark with other microneedle patches that were made before using other materials, they are comparable.
Yeah. Well, yeah. I was going to ask, how do they compare against the standard fill and vac method? But then I realized the fill and vac method produces Swiss cheese or broken microneedle. So it's kind of the benchmark. It's hard for you to benchmark because the benchmark wasn't really working, right? So
yeah, exactly.
As you've mentioned, you've got your microneedles through a release of 2 months. Mm-hmm. How did you measure the fact that it was able to release over 2 months? How would you go about doing that?
Okay, so in order to look at the release of the release profile of the microneedle formulations, what we do is we would do this. The very basic principle is that you expose the microneedles to a fluid. In our case, we use a phosphate buffer, which is I guess you could call it a simulated biofluid. So it's kind of, in a very simplistic way, it kind of simulates the fluid in the skin. And we will look at how the polymer behaves. So we look at how it swells. Again, we use imaging techniques a lot here. For example, use time-lapse imaging to look at how the polymer changes in shape. And in some cases, we can actually visualize the drug being released from the microneedles. So that's the first kind of investigation. And then we can actually kind of quantify how much of the drug has been released over a day, 2 days, or 3 days, or a week, or a month. And we do that in a small piece of apparatus called a Franz diffusion cell. The idea is the whatever's at the top, at the donor, will release the drug into the bottom chamber through the membrane in the middle. And this kind of simulates what the patch does on the skin, because the patch will contain the drug, the skin is the membrane, and then the drug gets released through the microneedles into the deeper layers of the skin, which is like the under, the receptor chamber in this Franz diffusion cell. And that's what we use. And the fact that you have got this compartment that is receiving this drug, from the microneedles, means that you can actually, if you take a small sample out of that, fluid, and analyze it using a technique like high performance liquid chromatography, you'll be able to measure exactly what drug concentration is in that chamber at any one time. And with that, you can build up a drug release profile.
And there's a couple of things you've mentioned. The first thing is that you would have somebody taking samples to run it using high performance liquid chromatography to build a release profile. Mm-hmm. And you would do that. The second point that comes to mind is over a range of time points. Have you ever found that having that sort of the experimental error mm-hmm. of that person taking a sample between either different people or on different days, has that mm-hmm. ever been an issue?
Well, I think it's always an issue with the manual sampling by a researcher. And it's not just about the error. I mean, the error improves with the competence of the person and the skill of the person, but you can only improve so much, because with human error, human error is basically random error. So you do have that error, but that error is kind of less controllable. So we did have that. I guess we live with it for a while.
Yeah.
But in this particular study, actually, we also wanted to address that, which is why we also developed an auto sampler to use with the Franz diffusion cell. And we call this Otto. So it's an auto sampler for the Franz diffusion cell specifically. And we developed it for a number of reasons. So when we started developing these ultra microneedles with a very prolonged release profile, we knew that we needed to sample this. But with the sampling regime is such that in the first 24 hours, you need to be sampling every so often. Inevitably, it means that the researcher has to come into the lab out of working hours. So, at night, at dawn, just odd hours, in order to take a sample from the Franz diffusion cell, just to make sure that you capture those data points in order for you to be able to build the correct release profile. And that was a challenge. And actually, we saw the challenge being amplified during COVID. So actually, one of the motivations for developing Otto as an auto sampler for Franz diffusion cells was we realized that during COVID, our work was being held back because we couldn't get into the lab. For a lot of researchers couldn't get into the lab to work, and actually that triggered a thought in me that if you had a robot that cannot be infected by COVID, would it not be good to have the robot do the work? And the Franz diffusion cell study was one that we kind of targeted, and we kind of developed Otto for it initially. So the way that we did it was we basically, I perhaps should say that there are automated systems that you can use with Franz diffusion cells. For us, we had the Franz diffusion cells, but we didn't have an automated system to sample it automatically. So we then wanted to develop the automated system. But we couldn't afford the commercial automated systems because they're typically very expensive and they don't work with our Franz diffusion cells, because these Franz diffusion cells, they come in variations of sizes and designs and things like that. So you couldn't really retrofit an automated system to the Franz diffusion cells you already have. And if you were to buy the commercial product, then you have to buy everything, which is too expensive for us to afford. So we thought that we would develop our own, and we would start with something cheap, but that would give us a basis for basically a lab robot. And we realized at the time, because I had just got into 3D printing, and I realized that a 3D printer is a very efficient but affordable robot. So we adapted a 3D printer and made it into a Franz diffusion cell auto sampler. We call it Otto.
Okay. Yeah. So I think this is where things got very interesting from a reader's perspective. So when I was reading your paper on it, it was quite interesting to see how you've taken something. You've used a range of different things that you wouldn't expect to create this auto sampler. So let's talk a bit about the logistics of how this robot actually works, right? So we've discussed the idea that this robot is going to be taking samples that frees up the experimenter. They're not going to have to come in early dawn or in the middle of the night. How is it actually taking samples, and how does this work? How does the robot actually do its function?
Yeah, so actually in designing the robot, I mean, I knew that we couldn't make it too complicated. Yeah. Because with the industrial robots, what you have are kind of actuators and sensors and a lot of things that have to work together, and also work with firmware and software as a package. And we didn't have the luxury of being able to afford the resources that will be involved in making the software and the hardware. So we wanted a very simple system. It just so happened that with the Franz diffusion cell, as I described earlier, there's a receptor chamber and a donor chamber. But what I didn't say was there's a sampling port that's kind of sticks out at an angle from the receptor chamber. And actually, the robot makes use of that sampling port. It actually exploits a property of the sampling port, because when you fill the receptor chamber up, it also fills up the sampling port. But it effectively creates two compartments. So the sampling port is one compartment, and the main receptor chamber is another compartment. So with Otto, what we do is we put, basically, we connected. So Otto is basically the gantry or the motion system of the 3D printer that we used. Removed a lot of the parts that are not of the 3D printer, that are not involved in sampling from the Franz diffusion cell. So things like the hot end, remove that. And replace that with a parts that we will then use to sample from the Franz diffusion cell. So what are these parts? These parts it consists of a pump, so a peristaltic pump, some kind of silicone rubber tubing, some kind of needles, flexible needles, metal needles. We try different sorts, and the way that it works is that we will have the tubing connected to the pump, and on one end of the tube we have a needle, on the other end of the tube we have another needle, and we put this end of the needle into the sampling port of the Franz diffusion cell, and the other end will have to go into a Otto sampler vial for the high performance liquid chromatography. So these Otto sampler vials are standard equipment that is used for, any sort of high performance liquid chromatography or HPLC.
Yep.
So we didn't have to do anything extra about that. We just needed to make sure that the needle goes in to a vial on that end.
Yep.
And on this end, the needle will be in the sampling port, and it will basically sample, it will suck up the fluid thanks to the pump. So the pump will pump out the fluid from the sampling port through the tube into the Otto sampler vial. Okay. And so the mechanics is actually very simple in terms of how it samples. The challenges come in. So the first challenge is you're not fixing the tubing, the position of the tube, or where the fluid goes. It does not always go into the same vial. It needs to go into a different vial at different times. So that was the first challenge that we had with this very simple system. And the way we solve it is actually, this is where the 3D printer comes in, because the 3D printer has got a gantry. It allows, it can move the position of whatever you attach to it. It can move it in 3 dimensions, in the X, Y, and Z dimensions. So we actually use that gantry system to move that needle from one vial to the next one, and to the next one, to the next one at different time points. So on the other end, the other needles kind of remains in the sampling port of the Franz diffusion cell, and the movement of the gantry at different times. So we'd activate the gantry. Say if I want to sample at 1 hour, we activate the gantry at 1 hour, and the gantry will move the needle, insert it into the vial, and actually that very motion of inserting the needle into the vial activates a switch and activates the pump, and then it pumps the fluid out from the Franz diffusion cell into that vial. And after it's done that, it will move to another position and push some syringes. And these syringes were pre-filled the syringes with the simulated fluid, which is a phosphate buffer, and it will push a certain amount back into the Franz diffusion cell to refill it. So it will complete this sequence of motions, and this will reproduce the normal kind of the routine, the sampling and the refilling routine that otherwise would have to be performed by a human researcher.
You say, given time points, it takes the sample. What are you using to program its ability to take a sample at, let's say, hour 1 or hour 15 or hour 24? How are you programming this, or what are you using?
At the heart of it is some G codes. So G codes are what 3D printers use, and a lot of kind of similar systems like CNC machines use to control motion. So so any sort of gantry that moves in 3 dimensions, X, Y, and Z. A lot of them can be controlled using G codes. So G codes is a quite a simple language that these machines understand. We use the same language because because we have adapted Otto from a 3D printer which already understands the language. So we thought we would use it. So that's how we program it. You can write G code manually. It's human readable. So you can write this in a text editor and feed it to the printer, and the printer will understand it. But because it's difficult for humans to write code in general, we tend to make mistakes. And if we make a mistake, then the printer will not understand the mistake, and it may execute a mistake, or it may not work at all. So we wanted to make Otto easy to use, so without having to worry about writing code. So actually, I developed a piece of software which I called Automate. So this is a very simple program that's just written as a portable HTML file. So it's like something that you can open up in a web browser, any web browser, and it will be a graphical user interface that allows you to kind of type in values and point and click certain actions in order to generate that G code automatically. So all you need to do is to know what intervals you want to sample at, what starting positions you want the gantry to be at, what sort of volume are you trying to refill, and you translate this into values that you can input into into this document that's sitting in your browser, and that allows you to generate a G code which you can then download and then load onto the printer in order to execute those actions. So the Automate software simplifies that for, I guess, normies, who do not write code, and who find writing code difficult, but it kind of simplifies that workflow.
The fact that you've created a platform where you can write, the volume that needs to be taken, or the frequency and the intervals, and then that converts it into a code for the machine to kind of communicate with or to receive. I think that's a really nice way for normies, as you say, people that aren't in that field, to use it. And it's really important because what you've kind of done there is you've removed limits on people that don't write code to be able to still innovate. As in this case. So for example, if somebody has an idea, they don't necessarily have to be able to go and spend weeks learning on how to write code. They can just use Automate and use basic English to create your frequency, your sample volume. So I think that's really nice to have available for scientists and innovators in general.
Yes. I think that that was brought out of necessity as well, because at the time we were developing Otto, actually, I would say most people on the team do not know how to code. So it was out of necessity that I created the software to basically help the team do their work as well. But it is also a very good way to make it accessible to other people who are in a similar position, who don't want to just don't want to bother with writing code and things. Who can just want to focus on the pharmaceutical research.
So how did you validate this, right? So in terms of not only how did you validate it, but how does it stand up against the conventional of a scientist going and taking samples in terms of error or percentage errors and sort of frequency? How did it play up against regular sampling?
So in order to validate this, we set our benchmark. Well, we set a human researcher as the benchmark. So actually what we did was we benchmarked it against a human researcher doing the work. So we had, at one point, we had a researcher doing this who was sampling alongside the robot. Now, because the researcher couldn't come into the lab at night or at dawn, they didn't sample at those time points. So they did the best to kind of cover all the time points they needed to cover during the working hours, but they had to miss those time points, which would be difficult for a human researcher to cover. And that was kind of part of the point as well, because realistically, if a human researcher was doing this, they would have to miss those time points. And they were doing this alongside Otto, sampling around the clock. And that was our benchmark. And what we found was that Otto, at different stages of development, showed improving consistency compared to the human researcher. In terms of the consistency, what we mean by that is because of how Otto works, as I mentioned earlier, the consistency hinges a lot on the sample volume, or the volume of the phosphate buffer, the simulated biofluid that gets refilled into the receptor chamber of the diffusion cell. And that basically dictates how consistent the sample volume is. And the sample volume then directly impacts on the accuracy of the data. So what we found was the consistency with Otto, as we kind of kept improving Otto, a lot of that improvement work was basically making sure that the screws are tight, you know, which part of the needles and the vials were aligned. So we have described, you know, some of these optimization work in the papers, but this work took months and months to dial in all these parameters. But we could see, you know, as we were doing this work, that the consistency was improving. And at some point it overtook the human researcher. So the human researcher was doing the sampling, and obviously there will be inherent errors, you know, random errors associated with human operation. There were also random errors associated with the autosampler, but the autosampler, we could see, you know, its consistency improve. So we were able to kind of reduce the random error over time as we kind of dial in all these parameters. So yes, eventually in its most recent iteration, Otto actually performed better than the human researcher in terms of variability of the sample. So it was more consistent in terms of the sample volume and also the accuracy. Another big thing that we found as well with the human researcher not being able to come in at night or at dawn to take a sample is that if you leave the experiment running for hours and hours without taking a sample, eventually that itself will impact on the quality of the data that you get, because of this phenomenon whereby the drug keeps kind of getting released into the chamber, but it's not getting removed during sampling because you're not doing sampling.
Yeah.
And so that drug accumulates in that chamber for a long time. And what that does is it reduces the concentration gradient. So basically the difference in drug concentration within the receptor chamber and within the microneedles, that difference is getting smaller and smaller. And that difference is what drives drug release. So that is the diffusion gradient, right? The concentration gradient. So if that difference gets smaller and smaller, there's less drive for the drug to get released. And therefore you would see apparently the drug release slows down. But that is an artifact. It's not a true reflection of what would happen in the skin, where the drug gets carried away by blood flow and things like that. So we found that this artifact becomes apparent with the human researcher who didn't sample overnight, but with Otto, it completely eliminates that. We didn't see that with the auto sampler that was able to run unattended, you know, overnight.
Was this the first time you've done something like this? So I know you've obviously done the Vac and Fill, but this is very different in terms of, you know, you're using a lot more computer involvement. Was this the first time you've done something like this?
Yes, actually, it's been quite a interesting learning journey for myself as well. I guess for me personally, I think the reason that I haven't done this before is because the tools weren't available to me before this.
Okay.
And actually, a lot of these things became possible because of 3D printing. 3D printing became available, and it just made things easier to prototype. You know, when you're working on innovating on the formulation, you will at some point encounter requirements for your formulation that are not met by current technologies that you can buy easily off the shelf from suppliers. Or so at some point you have to develop your own kind of bespoke technology, and that's what we have done here. But it has been very difficult to do that before technologies like 3D printing became available. And actually, we have been doing that for a long time, trying to solve these lab research problems that we had, but we always resorted to duct tape and masking tape and blue tag. Things are not very precise, but it kind of cobbled together something that kind of worked. But I think with 3D printing and the technology, robotics and things like that, it just allows us to build a bit more precision into these solutions that we kind of cobble together. So the solution becomes a bit more reliable, I guess.
Yeah.
Yeah.
What about this journey of creating Otto has surprised you the most?
What has surprised me the most? I guess, from concept to implementation, it has evolved in some ways that I did not expect. When I started building Otto, I didn't have in my head like the end product, like the fully assembled end product. So that has kind of evolved over time. Because I didn't anticipate, as I said, a lot of the details are, the devil's in the details. And because a lot of the details were not clear when you first start out, and you don't see those. I guess what has surprised me is how long it took to get it to where it is. Because a lot of the times I thought something would definitely work, but it's the things that you least expect that kind of threw you off. And a lot of things are very mundane. Like I said, trying to dial things in is a matter of tightening some screws that we overlooked. So that kind of made the development, it prolonged the development, I guess. I didn't expect how long it would take, but it did take a long time.
How long was it from sort of idea to final product did it take?
About 4 years.
Wow.
So I think the idea came about around the time of COVID. As I said, the COVID kind of motivated the idea to some degree. But we came up with, I think we published the first paper in 2024 on the validation. So we still keep iterating, to try to improve it and make it easier to use, make it more reliable. So it's not to say development is stopped. We're trying to make it better.
Is there anything that you want to bring up that maybe I've not asked, or something that that was on your mind?
I guess maybe a message to my fellow researchers. Don't be afraid to tackle problems that are slightly outside of your kind of main research focus. For us, I think a lot of people will look at the development of Otto Vac and Fill techniques as being slightly outside of what their focus is. Like if you're focusing on developing microneedle formulations for prolonged release, then you may not be interested in this. You may think that this is actually not worth my time pursuing. I guess my message would be, don't be afraid to try these things that are maybe slightly outside of your expertise. Because the lesson I've learned from this is that, coming with these solutions, they are themselves problem solving. This solves some challenges for us, and actually it's enabled us to actually pursue the thing they want to pursue a bit better as well. Because with the automation solution that we've come up with, we can now actually run experiments without these huge gaps in the sampling. Ultimately, I think it benefits what you are focusing on as well.
No, I think that's a very nice message to end with, but also it summarizes, it links quite nicely to your philosophy as well, which you can see from your work, is that you do venture out quite a bit, with both of the major works we've discussed. Firstly, even with the long-acting injectable microneedles, you've tried to go above and beyond what's conventional, and then you've gone with the sort of Vac and Fill, which is again trying to overcome a conventional issue, and then the testing, again overcoming that conventional way of testing. So it matches quite nicely with what you're saying and what your philosophy is. So thank you for saying that. And I just want to thank you as well for taking the time to join me. We've been going for an hour and a half now, so we'll start to wrap it up. But thank you for joining me, and honestly, it's been a pleasure to speak with you. And thank you.
Thank you again for inviting me. And actually, again, it's also my pleasure to speak to you about this and actually share this with your audience. Thank you.